Okay, welcome back to the last screencast for this module. We're going to jump around on a few different little topics, but to start with, we're going to look at how knowing what we might know about alleles and genotypes and that sort of thing, how we can actually predict what the offspring or likely offspring will be from two different parents. So we're going to look at something called Punnett squares. Punnett squares were invented by Reginald Punnett, so they're actually named after him. And they're a way of mapping out different genetic crosses and their possible offspring. So one thing to remember here is a Punnett square is just like what might happen. It's not this will all definitely happen. It's just if we've got two parents, a mum and a dad, and we combine them and they're going to have a baby together, be that human, animal, plant, or whatever it is, then for that baby, these are all the possible options that those parents could produce in terms of the genotypes that they could make, and then you can also work out the possible phenotypes from that. So these are what we call genetic crosses, where you have two organisms, they have specific traits, we breed them together, and we record the traits in their offspring to actually see what's happening there. So it is key to remember that here you are just doing, it's a probability thing. There's no certainty that this will happen, and you would have to repeat these crosses or these Punnett squares essentially for every time that you breed these two parents together. Although technically, once you've done it once, it'll be the same probability for every subsequent one of their offsprings for that trait. So over here, you can see our father here, he has a big B and a little b. Those are his alleles, so that's his genotype is big B, little b. And mum also has the same. So she also has big B and she also has a little b. So what we do is we draw the alleles separate because the parents can only ever pass on one of their alleles. They can't pass on both of them. So dad's either going to pass on his big B or his little b. And same goes for mum. She's either going to pass on the big B that she has or the little b. So then we would go, okay, well, what possible offspring could they have? Now, this is just one example. The parents don't always have to have a capital and a lowercase combination in their genotype for their alleles. They might be both dominant, so big letter and a big letter, or they might be both recessive, so a lowercase and a lowercase letter. So here we have two heterozygous parents, and we're looking at what happens with them. So here you can see the dad has a big B if he passes it on, so we'll use this box to represent what happens if dad passes the big B on. So there's a big B there, and we'll also say mum's passing on her big B. So one possibility is you could have offspring that is big B, because they get those big dominant alleles from both their parents. You could have this one over here. Dad might only pass on the lowercase b, and mum might pass on her big dominant b. And that's actually kind of what's happening here. It's the other way around though. Dad passes on the dominant and mum passes on the lowercase. So both of these two, we have a big B from one parent and a little b from the other parent. So you end up with a heterozygous offspring. Not definitely will. Again, this is just one option that you could end up with. And lastly, you could have dad might pass on his little b and mum might also pass on her little b. And so we end up with offspring that is little b, little b. So this would be homozygous recessive here. And then you can go through, if you know what the phenotypes are, you can go through and work out what phenotype should be expressed for each of the possible genotype. Now, this doesn't mean that if these two parents have 4 kids, that you will get one of each kind. It just tells you that they have a 1 in 4 chance of getting big B, a 2 in 4 or a 1 in 2, a 50% chance of getting big B, little b, and a 1 in 4 or 25% chance of getting little b, little b. So every time they have a kid, these are the possible outcomes that that kid could be. So that's what a Punnett square is. On the next slide, there's a little animation that's going to play. So just watch what's happening there and you should be able to see the information get put in and also fill in the Punnett square for that cross. So here we have some guinea pigs and we've got 2 heterozygous parents and we're just using their information to fill in the Punnett square. And then we can analyze it from there. So you can see here, this time around, we've got a homozygous recessive parent. So it changes what's happening. All right, so if you follow this, you can see the parents' genotypes are put up the top with 1 letter above each column or to the side of each row, depending on which parent you're talking about. And you then take their information and you fill it into the boxes beneath. So all the boxes in this column will have a capital H from this capital H, and all the ones in this column will have a lowercase h from this one. So that's how you fill that in. And I know this keeps changing, but the idea is still the same. As you go across, you then also fill in the letters there. So this row will all have lowercase h from this one and the same from there. It'll all have from here. What you'll notice at the end is once all the cells in the table are actually filled in, it's then going through and analysing, okay, what do we actually have here is present? So this time around we had homozygous and homozygous, 1 dominant, 1 recessive and crossed. So there are 3 examples of Punnett squares happening here. And so here we've got our thing finalised and then it analyses it and it colour codes it based off the different types of genotypes that you've got there: homozygous dominant, homozygous recessive, and heterozygous. And it looks at the percentages of each. So generally with Punnett squares, you need to determine your parents' genotypes. Okay, you can't go anywhere really without knowing the parents' genotypes. There are sometimes you might get a question where you will know all the offspring or the possible offsprings, and so you might have to engineer it backwards to work out what the parents were. But most of the time you need to start with the parents' genotypes. You're going to add their alleles to the Punnett square. So one parent will have its alleles at the top of each column, and one parent will have it to the side of each row. It doesn't matter which one's the father and which one's the mother. There's no rhyme or reason to it. You just pick one for one spot and one for the other. Make sure one letter gets written above each column and one letter beside each row. Do not do two. So this parent has big H, you wouldn't write big H above each column. You'd write just one big H before the column, above the column. It's important that you don't actually combine the two alleles. You need to separate them because they can only pass one on. Then you fill in the Punnett square like this animation's been doing with all the different possible combinations, and then you can start to calculate probabilities of the genotypes and also of the phenotypes. So let's go through an example together. So here we're going to first determine our parental cross. So our parents, one is big T, little t, and the other is big T, big T. So we need to set up our Punnett square with their alleles, and that can go outside the box. Sometimes you'll see Punnett squares drawn where you'll actually have boxes around the edges. You don't have to draw it that way if you can, if you like. So our first parent is big T, little t. So the big T goes above one column and the little t goes above the other column. Then the next parent is big T, and it's going to have its big T. One of them will be next to one set of rows and the other will be at the start of the other set of rows. Then we need to fill in the Punnett square to determine the genotypes for their offspring, all the possible ones. So this is what's going to go in the boxes. So this one's going to get a big T from here and another big T from above. Then the next one's going to get a big T from, because it's in this row, but it's going to get a little t because it's in the little t column. And you keep filling that in. Now this has it written with the red ones in front every time. For the ones that are both big T's, it doesn't actually matter. It does matter where it says big T, little t, but not because of the colour, but because the big T has to go first. Here where it's big T, you could have it the blue one, then the red, or the red, then the blue. Normally you just write it in the one coloured pen. This is just showing you where those letters are coming from. That's why they're different colours. The order in that case doesn't actually matter. It only matters when you're talking about the capital and the lowercase. You always put the capital first if you've got both. Then we're going to determine genotype and phenotype probabilities. So starting with genotype, you'll notice here that half of them are big T, and half of them are big T, little t. So 50% are big T, and 50% are big T, little t. So that's your genotype probability. Phenotype probability, all of these, okay, have a dominant allele in them, which means they're all going to express that dominant phenotype. In this case, the dominant phenotype is round P's. So in this case, 100% of the offspring are going to have round P's. So even though you might have half of them end up having a heterozygous genotype and have the recessive trait in their genes, they won't express it because the dominant one is also present. So all of them will actually express the dominant phenotype, which is round P's. So we're actually going to practice doing Punnett squares. And we've got 4 examples here for you to go through. You need to follow the steps there laid out on the left. You need to work out what the parental cross is. So you need to work out the genotypes of each parent. You then need to set up your Punnett square. You then need to fill in the Punnett square in the boxes in the middle to work out what your offspring could possibly be. And then you need to work out the genotypic and phenotypic ratios, or the percentages. So you can pause the video and go off and do this, or you can do this after the video. This will be a separate thing. You'll need to submit your work to the teacher to show that you've done it. So just look out for the submission spot for this activity. Otherwise, we're going to keep on with the last little bit of the PowerPoint. Okay, so a few last little points before we sort of wrap up this module. First of all, when we're talking about across, your parents in it are called the P generation, P for parents. So you can see that on the side here. So we've got 2 parents. When they combined and they are bred together, they're going to produce a generation bunch of offspring. That first generation they produce are called the F1 generation. You can see here where the name sort of comes from. You don't need to worry about it. We just call it the F1 generation. If you take 2 members from the F1 generation and you breed them together, so yes, they would be siblings, you can breed them together, and then you get a second generation from that. And this is called the F2 generation. So that's what this is. So if you see P generation, F1 generation, or F2 generation appearing, that's what it's referring to. It's just a way of labeling the different generations in a genetic cross. So far we've talked about something that we call complete dominance, where you have generally 2 alleles and one is instructions on a phenotype that's completely dominant over the other one. Sometimes that doesn't actually happen. And one of the main ways is we see this happening where you sort of don't have necessarily one allele being dominant over the other is something called codominance. So this can be there's more than one dominant allele. Sometimes there's still a recessive one. Sometimes there's no recessive allele. And so this is what codominance is. And this is interesting because with complete dominance, your heterozygote will express only the dominant trait. But in codominance, because you have more than one trait being dominant, it has to express both of them. So heterozygotes in codominance will express both phenotypes. So chickens here are a bit of an example. You might have with feather color, white is one type of feather color, black might be the other, but you might find that they're both dominant. Now this is a scenario where you do actually, for the same gene, use different letters because they both need to be capitals. And so you can't use a capital W for both the white and the black one. It'll get too confusing. You won't know what you're referring to. So there's a few ways that we can overcome this. One is to use different letters to represent it. And so this is sort of one of the few times where that's acceptable. Normally, if it's the same gene, you use the same letter. So here, there's a bit of flex. Okay, so capital W would be the dominant white allele, and capital B is the dominant black allele. And so if you have big W, it's a white chicken. If you have big B, it's a black chicken. But in this case, if you have the heterozygote where you have a big B and a big W, you actually end up with a speckled chicken because some feathers end up being white and some end up being black. So you end up seeing both phenotypes being expressed. Some parts of the chicken are white, some parts are black. So it's all speckled all over. So that's what codominance is. That's one example. We'll look at another as well. So this other example is actually with humans. And this is also going to show you another way of representing the different alleles. And that's with blood groups. With blood groups, there are actually 3 different alleles that you can have. There are 2 dominant ones and 1 recessive one. Now, for all of them, we use the letter I. Okay, so the dominant ones use a capital I and the recessive one uses a lowercase I. That's fairly straightforward. That's like normal complete dominance, what we've been looking at this whole time. However, there are 2 different dominant ones. And so one of the dominant ones is a capital I and then a superscripted capital A. And the other one is a capital I with a superscripted capital B. And so that's how we represent the difference. We still get to use the same main capital letter here. And then we have a little tag almost saying, oh, this is the A version of it or the B version of it. And then, of course, we still have our little I, which is representing the recessive. And so what happens here is if you have IA, it makes something called antigen A on your red blood cell. If you have IB, it makes antigen B on the red blood cell. If you have a little I, it doesn't make any antigen on it. Okay, so this is sort of what we're talking about here. So I think antigen A is like the blue circles, B is like the purple cones, and if you have little I, it doesn't make either of them. And so what can happen is you can have all these different combinations. So instead of the normal 3, we can actually get 6 different combinations. If you have IA, you can only make antigen A. So you have what we call blood type A. That's your phenotype. If you have IB, you can make both the A and the B antigens. So you have what we call blood type AB. That's your phenotype. If you have IA and then little I, that little I, it's recessive, you wouldn't express it. So you end up just expressing the IA, which means you make the A antigen, so you have blood type A. If you have IB, then you're going to express that dominant one. You're going to make antigen B, you have blood type B, and that's your phenotype. If you have IB, it's pretty much the same here, like we were talking about with IB. Same idea, you've got the recessive one there, you're not going to express it, you only express the dominant one, and so you end up making antigen B, and so you end up with blood type B. If, however, you have both recessive alleles, so little I, you won't actually make any antigens on the surface, no little bumpy things here. And so you actually end up with what we call blood type O, which is really meant to be blood type like 0 or 0. There's nothing, no antigens being produced, and so that would be blood type O. So that's how we get all the different kinds of blood types. We have A, AB, B, and O. That's just one way we can also have codominance expressed there. Now, just before I finish, you may sometimes see a type of dominance called incomplete dominance. It's not something we're focusing on in this, but in case you're interested, incomplete dominance is often just talked about as you have 2 dominant alleles, and when you have a heterozygote, it produces a phenotype partway between the two. So it'd be like if you had the chickens, it would be like having the white and the black. Instead of producing a speckled chicken, would produce a grey one because it's halfway between white and black. But that's not something we're really getting into or we're covering in this unit, so you don't actually need to worry about it at all. So if you see it somewhere, you can just dismiss it. Anyway, that is it, and that brings us to the end of the screencast for this module. Don't forget to go back and do those Punnett squares if you haven't done them already. All right, that is it. Bye for now.